contraction starts with a neuromuscular control. The axon of the nerve cells of the spinal cord branch and attach to each muscle fiber, forming a neuromuscular junction. An action potential passes down the nerve. The nerve releases calcium and the results into the acetylcholine. ACH binds with the sarcoplasm and where the muscle fibers attach action potential. ACH binds with the receptors and opens the NA channels. NA channels open and the comes in. When the NA goes in, there is a decrease in the resting potential. NA rushes in and the sarcoplasm depolarizes. The regional depolarization spreads rapidly. The positive patch in the membrane changes the adjacent patch of the membrane. Thus, depolarization spreads. The K channels open and the region repolarizes. Immediately after the action potential passes, the membrane permeably changes again. NA channels close and K channels open. K rushes out of the cell and cells repolarize. Calcium is released from the sarcoplasmic reticulum. The calcium is stored in the sarcoplasmic reticulum, which depolarizes, releases the calcium. The calcium clears the actin binding sites. During the muscle contraction, the thin actin filaments slide over to the thick myosin filaments. When calcium is presented, the block active sites of the actin clears. Myosin head attaches to the actin using high-energy ADP and phosphate configurations. Power strokes are used. Myosin heads pivot, pulling the actin filaments toward the center. The crossbridge detaches when a new ATP binds with the myosin, which then cocks the myosin head, occurring when the ADP, and phosphate into another crossbridge cycle.